Hello everyone, this is Mrs. Travai and this is the Introduction to Research in Molecular Biology course. We are going to be talking about vectors and libraries, and this PowerPoint has been provided by the Waxman Student Scholars Program at Rutgers University. So let's take a look at what we're going to be learning. Chapter 1. We're going to be looking at recombinant DNA technology and how we are able to manipulate genes to be able to study them. And it's been for many years, scientists have tried to engineer or try to analyze genes, and it's been very difficult. It's been frustrating, and in some cases, some scientists dedicated entire careers to purifying just one gene. Genes are made up of nucleotides, and they are found on chromosomes, and we're going to take a look at that. So how do we actually study or purify individual genes? In the past, some of these names, these people here, are important information, important figures. They actually were given a Nobel Prize for genetic engineering experiments, and they are the foundation of what this research, or how this research was built. We're going to be working with a plasmid called P-triple-EX, and that is going to be our vector. And what Rutgers has done for us is they inserted a gene into a vector and placed it and amplified it using bacteria. This is a much less expensive process if bacteria are grown and amplify it naturally. Vectors are found naturally in bacteria, and we took that information, scientists took that information to be able to understand how to insert a piece of DNA into a vector and be able to transfer it into a bacteria or transform it into a bacteria such as E. coli. It can also be a virus. There are other ways to transfer vectors. Plasmids, and this specific plasmid that we're going to be using, is a circular piece of DNA found in bacteria. The host bacteria replicates it. Basically, if it has an origin, it binds to the origin, and that will be the site of replication. And in most cases, bacteria such as E. coli may have even up to 500 copies of a plasmid in their cells. This is great because that gives it a large volume per cell, and when those colonies multiply, or those cells multiply, they form large colonies. This plasmid also has some very important portions or sections. In one section here, we have what's called the Lac Z gene, and this Lac Z gene actually allows us to select for the bacteria that have an insert in this multiple cloning site. The multiple cloning site, the MCS, is actually a section of the plasmid that has many restriction enzyme sites back to back, and they don't write them in here because there's so many. Your insert is actually inserted in this region and will disrupt this gene. This gene allows the bacteria to digest a molecule similar to lactose, and it looks a lot like lactose, it has same binding sites. And when it digests it, it does not allow, it will actually, the bacteria, if it produces this protein, it can digest the X-gal that's on your Petri dishes and turn blue. When we insert a gene into this region, we disrupt that protein, we disrupt that gene, and we may only make a partial protein which is not able to digest the X-gal on the plates. So those bacteria will not turn blue. In addition, we use another selectable marker, the ampicillin resistance gene. This ampicillin resistance gene produces a molecule which will break down different types of will break down antibiotics, specifically ampicillin. There are different types of antibiotic resistance genes that can be placed in different plasmids, and in this case we're using ampicillin. So how efficient is transforming bacteria? It's not very efficient, surprisingly. We do this in the lab in AP Biology, where we take bacteria and we introduce a plasmid that allows the bacteria to glow or to fluoresce. And in this case, we're not doing that. We're going to be transforming a plasmid. Rutgers transformed a plasmid into the E. coli that carries your insert. So this, basically, the ratio is about 1 to 1,000 cells except the plasmid. So how do we make sure that a plasmid enters the cell? First, we need to treat the cell with calcium chloride and shield the negative charge from the membrane so that this negatively charged piece of DNA can actually slip through its pores when it's heat shocked, or they can also use something called electroporation. In this case, the plasmid goes in, but for our purposes, it's good that actually only one will get in. That way, we know that only one gene is in your plasmid or is in your bacterial culture. So what happens to the other bacteria that don't get transformed? Well, basically, they don't have the plasmid that allows them to break down ampicillin. All your culture material, all your petri dishes will have ampicillin in them, and therefore, when these bacteria don't accept it, the plasmid is only going to reproduce for the one that has the bacteria will only produce with the one that has the plasmid. Okay. And so in this case, that's what will happen. We have a bacteria that now everything else has died and it can survive. And on the petri dish, it will create a colony of bacteria with hundreds and hundreds of cells. And this is what it will look like. So the colonies, when the library is created at Rutgers University, they plate them onto petri dishes, which are called libraries. And in this case, you'll see that these cells that are growing must have the plasmid, because it actually allows this plasmid allows it to be resistant to ampicillin, allows it to grow. The dead cells don't grow. Some of the safety features with working with these types of plasmids is that they won't reproduce in nature. There's a level of biological containment that these engineered plasmids are unable also to transfer DNA from one to another. So we're able to do that to protect nature. But in naturally occurring bacterial populations, bacteria can transfer DNA from one to the other naturally. And this is a big problem in medicine, and especially in hospitals, where we might have a patient having an antibiotic-resistant bacteria, passing it to another patient through contact, maybe just the nursing staff or the hospital staff moving from one room to another. It could be as simple as that. And okay, this is the Lac Z gene. It produces beta-galactosidase. And I talked about it before. XGAL is placed on the plate, and this material mimics the lactose. And this beta-galactosidase linkage here is able to be broken down through hydrolysis, and you produce galactose and glucose. This is a form of nutrients that the bacteria can absorb, and therefore this happens naturally for bacteria that have the Lac Z gene. But in our case, when they break down XGAL, when this is actually cleaved, it allows the bacteria to turn blue. It creates a pigment that actually changes color and allows us to detect it. So basically, wherever you have the Lac Z gene intact, you'll have a blue bacteria. Wherever you have an insert or a gene of interest, such as the ones from our plant, in here, it disrupts the Lac Z gene. You only make a partial protein, and therefore the partial protein cannot digest XGAL and does not turn blue, so they are white. So we are looking for white colonies. Okay. And now, like I said before, our petri dishes that we receive from Rutgers are considered DNA libraries. They are basically collections, random collections of DNA that have been cloned into a vector. And ideally, it contains one copy of every DNA sequence that we have. So across the board, we may only get 4 library plates, but the entire library is plated out for participants in the Waxman Student Scholars Program. And therefore, hopefully, we are sequencing the whole organism by using all these plates. You can make libraries for genomic DNA or cDNA, which we'll talk about in a second. So in the construction of a genomic DNA library, which is done when you're sequencing huge genomes, you can use a process called shotgun sequencing. There are other processes, but this is just one of them. And again, it's the same type of process where instead you're starting with a chromosomal piece of DNA. You're partially digesting it to fragments. Those fragments are then being ligated into plasmids that have been cut with the same enzymes, so the sticky ends match. This is then transformed into bacteria. They replicate, and we're able to generate a plasmid or a library of bacteria that have different sequences or different genes in them. we pick random clones and purify them to be able to identify or send them out for sequencing. Once we sequence these inserts, then if you're working with a genomic library, we can ask, there are computer programs, or you can physically try to build contig sequences between them. In the case of the information, some of these sequences, like this blue sequence here, may overlap. It may match this green area, but it also matches this orange area. So when the computer program analyzes the pieces of DNA, it will tell us that this might be an overlap. And therefore, we eventually can actually completely sequence or provide the complete sequence by patching these all together. So the human genome initially cost about $3 billion to make. Nowadays, you can sequence at $1,000, depending on how much data you want, or how much or where you're getting a sequence. It might even be less than that. I've seen prices less recently. We're not working with a genomic library. We're working with something called a cDNA library. So what's the difference? Okay, a cDNA library is a copy of RNA. Okay, usually a copy of mRNA. Remember that there is RNA that is first produced during transcription that does not have a poly A tail, but it is processed, and later we call it, once it's processed in the cell, we call it an mRNA. And this is the DNA that we are using, or this is the sequence that we are using to form our cDNA library. So this picture here is very important. This is something that I will refer to often, not only in the actual lab work portion of this course, but also in the bioinformatics section. And the DNA, you should know the different parts of a structure of a gene. You should know that all genes have a promoter region. They all have a start, an open reading frame, a stop codon, and an area where there is a non-coding template on either side. And when this is transcribed in the cell, RNAs are created and they're processed to include this poly A tail. So now we see that we have this untranslated region and this other untranslated region included in the mRNA. When translation occurs, it will actually take the sequence, the ribosome takes the sequence, the mRNA sequence, and through translation will form a protein that starts with a methionine. And again, all proteins start with methionine and they end with a stop. All starts of the proteins are the amino end and the ends are the carboxyl end.